I'm so happy that you're here today, Dr. Anderson. I am Dr. Jo Bichell. I am a neuroscientist. I'm also the mother of a son with Angelman syndrome. And I'm really happy to be bringing the Angelman Updates to our families. It's a co-production of the Foundation for Angelman Syndrome Therapeutics and the Angelman Syndrome Foundation. And we're just trying to let people get to know all the science and the scientists a little better. So thank you so much for being willing to do this. And if you could just introduce yourself, let us know where you are right now and just a little bit about where you come from and what you're doing right now.
So my name's Jo Anderson. I'm an associate professor at the University of California, Davis in Sacramento, California at the Medical Center. My research involves developing stem cell and gene therapies for various diseases, including HIV, and some orphan diseases, including Tay-Sachs, Angelman syndrome, and some other ones that we're working on.
So how long have you been doing this?
Since graduate school, 2002, so almost 20 years. Yeah, I went to undergrad in Wisconsin and started working on molecular virology then, and then went to graduate school in Colorado and got into a lab that worked on HIV and gene therapy. And that's what got me into that field, learned about stem cells, learned about animal models, and then carried that through out here to Davis, and then used that technology for the other therapies that we're working on, the genetic therapies such as Tay-Sachs and Angelman syndrome.
So your particular form of genetic therapy is really kind of different from everybody else's. And it's from bone marrow, if I understand it right. And it makes it unique and really, really special. So could you talk a little bit about that? Why did you do it that way exactly? And what's good about that?
Yeah, so as I mentioned, the original work that I was involved in was an HIV stem cell gene therapy approach. And that uses the blood stem cells, which will create all the immune systems in the body, which HIV then infects. So we're able to genetically modify a patient's own blood forming stem cells to then protect the body from further HIV infection. So what other groups had done was they had used the same stem cells, but instead of genetically modifying them to protect the body from an infectious disease, they were using the immune system to deliver therapeutic proteins that were missing in the body. So now we're using the immune system as sort of a factory to produce and then deliver UBE3A for Angelman syndrome.
But the Angelman syndrome is not a blood disorder. I can understand how it would work with HIV, but how does it go into the brain and get into neurons, which is what we would care about with Angelman syndrome?
Yeah, so we aren't directly correcting the neurons. So we're using the immune system and the immune cells that do get into the brain, such as the microglia, to produce wild type functioning UBE3A, where the neurons will then take that protein up and it can function in a normal manner and hopefully correct the neurons that have that dysfunction.
Okay, so let me get this straight. So you take out the person's own bone marrow, the person who has Angelman syndrome. You put back in the UBE3A. Then you put the bone marrow back into the person. and then their own bone marrow makes cells that can cross the blood-brain barrier. Am I right so far?
Correct, except for the bone marrow part. So we wouldn't have to go into the bone marrow and extract the cells. We can give patients a drug that will push those cells out into the blood. So we can just take the cells from the blood instead of having to go into the bone marrow. But you are correct that the cells do come from the bone marrow. We just don't have to go into the bone marrow. We can take them from the blood. But yes, everything else is correct. Yeah.
Okay, so now let me get this part straight. So now the cells cross the blood-brain barrier. They're carrying a cargo of UBE3A, and they have that cargo. How does the UBE3A get into the right place at the right time?
So we have modified the gene and the protein a little bit to do special things that it normally wouldn't do. So yeah, so that's how it happens.
It's a little bit proprietary.
Yes.
So, but the point is that the UBE3A gene somehow, once it's actually in the brain, will actually go into neurons where it's supposed to be and into the right part of the neuron to do what it's supposed to do, even though it's being delivered by microglia, or, which is the form of a kind of a blood cell.
Correct.
Wow, that's amazing. And then if that happens, so do you have to keep doing the infusions over and over again, or how do you does the person with the Angelman syndrome just get one sort of infusion, or they need to do it for the rest of their lives, or how does it go?
No, so these stem cells you have as a baby, you have as an 80-year-old, and they constantly make the immune system cells in your body for the rest of your life. Like the red blood cells, the T cells, they have a half-life, they eventually die off, and your body relies on these specific stem cells to then make new cells for the rest of your life. So theoretically, you would just need to get this cell transplant once, and because we're modifying those stem cells. If we were to modify some of the mature cells, like if we were to take the microglia, gene modify them, and then transplant those, they would eventually die off, and then we would have to repeat it. But because we're doing a stem cell transplant, those stem cells will continue to make the UBE3A expressing immune cells for the rest of a patient's life.
Wow. So where are they sitting, those stem cells? Are they sitting in the bone marrow at that point after you put them back in?
Yeah, they know. So we would just do an IV infusion for the patients, and these cells have specific markers on their surface that helps them migrate and home back to their bone marrow spot, so they know where to go and where they like to be. So they will go back to the bone marrow and they will sit there and do what they have to do
For the rest of the person's life.
Yeah.
Unbelievable. And now, how do you know that you're putting in the right amount of UBE3A? People worry about too much UBE3A. You could be like, do 15 or Prader-Willi. I mean, how do we know that you're putting in the right dosage?
Yeah, so that is one of the tricky parts, because each individual person's stem cells will get gene modified at different levels compared to another patient's. And so what we're doing preclinically is looking at the different levels that we see in the animal model that we're using. And so we can look and see how much engraftment of our gene modified cells are in the animal model, and then we can compare those levels of engraftment to the behavioral and motor assays that we run the animals through and see if they act like, say, an overexpressing UBE3A with 2 copies of the gene, and compare those characteristics to the animals that that we're transplanting and looking at for the preclinical studies. But yeah, that is one of the difficult parts, is being with our system, where it's not a drug where you can just give a person a specific amount of that drug. It's all going to depend on how well their cells get gene modified. Yeah.
So has this been, no, I know it hasn't been tried in humans with Angelman syndrome yet, or wait, no, it hasn't. But has it, has the same idea that you have with the correcting the bone marrow, has it been tried in humans with other disorders yet?
Yes. So other disorders like adrenoleukodystrophy and metachromatic leukodystrophy, those have been in clinical trials. So people can look up that information.
What about any brain disorders?
Not that I can think of using this method.
So we would be the first.
Potentially, yeah.
Wow. And what, why did you get, how did you get sucked into the Angelman field?
I used to teach a stem cell and gene therapy class, and one of Dave Siegel and Kyle Fink's grad students, his name is Peter Dang, he took my class, and he came up to me afterwards, was telling me about his Angelman project. And then I told him about, why couldn't we, because it was a single gene disorder, why wouldn't we be able to do this for Angelman's? Because honestly, I hadn't heard about Angelman syndrome before that. But we were working on Tay-Sachs, and that's a single gene disorder for Tay-Sachs and or Sandoff disease. And so I was like, well, why couldn't we do this? And so we thought about it a little bit, talked with Dave Siegel, talked with Jill Silverman, and then we, applied to FAST with the idea, and everything's worked out great since then.
Wow, that's so cool. So let me ask you, you've tried it in animal models and definitely in mice already. And so what happens if you do this in an already grown-up mouse? Do they do anything better? Are they able to, or does it have to be done in a newborn, or how does it work?
So we perform the experiments in both ages. So newborn before symptoms developed, and then adults after symptoms start to develop. And we see wild type phenotypes in both cohorts. The younger mice don't develop nearly as severe phenotypes as non-treated mice. And then the adults sort of revert back to wild type phenotypes. So we see that it can help both ages. We haven't done anything later. I don't think we will need to for a clinical trial to start. That would be more of something like if we were interested in doing it, to do even older mice, but we haven't done that yet.
You mean like elderly mice?
Yeah, correct.
Oh, wow, that's just amazing. And so when you say they revert to wild type, do you mean things like motor skills, or do you mean things like seizures, or do you mean things like learning and memory, which is the holy grail of Angelman syndrome?
So we looked at learning and memory, and we looked at motor, but we will be doing the seizure experiments. We haven't done them yet. That was one thing that we were asked to do, so we will be doing those. But yeah, the mice in these experiments look similar to wild type, and unlike the non-transplanted or non-treated animals.
My God, this is just like a total miracle. I mean, you found a way to get UBE3A across the blood-brain barrier and manufacture itself for free for the rest of the animal's life. It's amazing. And it reverses the phenotypes. I mean, like, how excited are you every day when you go to work?
Yeah, I'm excited to get this into a trial. We've just been asked to do more experiments, to not just enroll adults in the trial, but to also be able to enroll pediatric patients in the trial. So we need to do a couple more experiments to show a direct benefit for pediatric patients. But yeah.
Wow. So do you love your work, or do you just like can't wait to go home and have a cocktail? I mean, how does it work?
No, I love it. I wouldn't do it if I didn't love it. I get to bring my dogs with me to the office, so they're sitting there with me when I'm in there doing work, and yeah, it's great.
There's a dog there right now, right?
2 of them, yeah.
What kind of dog?
They're Cavalier Spaniels.
Oh, right. Oh, I've heard about those kind. Can you show us one? Or are they too heavy?
Monster beads. Monster beads. Okay.
Let me see.
Can you guys sit?
Oh, are you at work right now? Is that where you
No,
are?
I'm at home. I'm doing stuff on the computer today.
Yeah. Well, that is absolutely amazing. I gotta tell you, I'm so excited about this work. Is there anything that families can do to help get this to, get to clinical trial?
I think it would just be advocates for us and for the trial. The FDA is our next hurdle to get the IND, and then we would be able to, start the trial. That's the next hurdle. We've shown that it's worked in the animal models. We know we can produce the therapy in our GMP facility here at Davis, and it's just a matter of getting it through the FDA to start the trial.
And how are the Tay-Sachs studies going? I mean, are they moving along just as fast as the Angelman ones are?
Yeah, well, actually not as fast, because we were able to get the funding through FAST to do this stuff. So it's been a little bit slower, but they're both at the same stage right now. We're both at the IND enabling studies for both projects.
Wow, amazing. Last question.
Okay.
So what do you do for fun?
Play with the dogs, pretty much.
Okay. Like take them on walks, or do they go to the beach? Or I mean, you're in
Walks,
Texas, you don't really have a beach.
parks, the beach. They love the beach. They love the water. Just got a new paddle board, so I want to see how they do on that, because that'll be a little tricky. But we have some nice lakes and some calm water areas around here in the Sacramento area, so I'm gonna try and get them on the paddle board.
Well, thank you so much for this interview and for the work that you're doing. It's incredibly exciting, and I just can't wait to hear the next installment. We'll have to call you back and do this again in about 6 months.
All right, sounds good. Thank you.
Thank you. Thank you so much. Bye.
Bye.